Hello, everyone, and welcome to the Natural Areas Association 2021 webinar series. I am Maura McGinty-Kloss, and I am the Outreach and Communications Manager for the Natural Areas Association. And the focus of my role is to ensure that NAA consistently does what the organization does best, which is to share information among natural area professionals and researchers. And I'm delighted that you're joining us today. As you know, the purpose of NAA is not just to convey information, but to engage natural area professionals in topics of common interest. So I encourage you to participate in today's webinar. Throw questions into the chat. If you're brave enough and you want to unmute, we can actually have a discussion with the speakers when we do Q&A at the end. So I hope that you'll think about that. The mission of the Natural Areas Association, or NAA, is a national nonprofit. It's dedicated to the support and advancements of natural area professionals who protect the world's natural resources through science-based conservation, stewardship, and advocacy. We are a community who identify challenges, develop solutions, share critical findings, and adopt best practices in conservation, preservation, and the restoration of nature through networking and collaboration. We've been around for over 40 years, and we support natural area professionals through our peer-reviewed journal, which comes out quarterly, our stewardship and action workshops, which are hands-on and in person, and we hope to get back to those very soon, our state natural areas programs, which are now virtual, and while they are primarily to support people with state programs and that work in state programs, they are open to anyone. And our newly formed Science Advisory Committee, which is leading thinkers and researchers in the area of natural sciences who will be helping to identify emerging issues for us, our Natural Areas Conference, and of course, webinars like these. So if you are not a member of NAA, we do have the opportunity for you to join these webinars as you did by paying $29, but our membership is just $79. So if you come to even 3 webinars this year, you will have easily paid for your membership. We will be offering a 7-day trial to everyone who paid to join us today. So we encourage you to get on the website, check out our archives, and see if maybe some of our programming and materials aren't of assistance to you. So I just want to remind you of some webinars that are coming up. So we host our webinars on the 3rd Tuesday of every month. So on February 16th, we'll be hosting Project Budburst with Dr. Emma Ostrin. March 16th will be Current and Future Carbon Storage Capacity in Southeastern Pennsylvania Forest with Jessica Settlebauer. And then in April, we'll do Communicating the Importance of Your Work, which is being actually hosted by a communications professional. And the purpose of this webinar is really to help you articulate the importance of the work that you're doing to decision makers in grants and those sorts of things. So we're really excited about that program. And now I want to take a moment to let's get started, and I will introduce our speakers. So today's webinar, Toward Improving Pollinator Habitat, Reconstructing Prairies with High Forb Density, is being presented by 2 highly respected natural areas professionals, Pauline Drobney and Diane Larson. Pauline is a zone biologist with the Midwest region of the U.S. Fish and Wildlife Service. She received her MS in biology as well as her BS degree in biology and art from the University of Northern Iowa, Cedar Falls. Her career of several decades has been dedicated to learning, practicing, researching, and teaching tall prairie grass and savanna management and ecological restoration. Diane is a research wildlife biologist with the U.S. Geological Survey, Northern Prairie Wildlife Research Center. She received her PhD in ecology from the University of Illinois at Chicago and her BA and MA from the University of Colorado Boulder. Diane's research focus is plant and animal interactions and ecological restoration, and she is especially interested in the intersection of these 2 processes. So I will now turn it over to our speakers.
Okay, so we're going to get started here now, and the first thing I'd like to do, thank you for the opportunity to present these this webinar to all of you. It looks like we have a pretty good distribution around the country, but we do have a poll that Diane and I would like to start off with. And there's 2 questions. The first question, maybe Mara, you could start that first poll. What is your main role in restoration ecology? And we'd like to know if you're a researcher, a practitioner, or have some other role. So if you could answer that. And the second one is to know what area of the country is in your main focus. This will help us kind of know who we're working with primarily. And it looks like we have quite a few practitioners out there. That's great. So about 64% practitioners and 14% researchers, and then 31% other. And then what area of the country? Let's see what we've got here. What we are primarily looking at, mostly in the Midwest, but again, just based on what I was seeing from people signing in and with their name and their location, really from throughout the country, mostly in the Midwest. Okay, great, that's wonderful. All right, now we have a second poll. We'd like to ask a question, and perhaps that you could launch that second poll, Mara. And that one is, do you struggle with infusing species diversity into your prairie plantings? That one jumped up, the yes jumped right up there. It's something that I think is a common struggle for anyone who does this sort of work. So not uncommon. So it looks like we've got 68% say yes, 19, somewhere in there, no, and then 14% or so for not applicable. Okay, thank you. We can take that poll off. I'd like, first of all, to thank our cooperators and collaborators, Sarah Vacek, JB Bright, Rebecca Esser, Karen Visti-Sparkman, all of the U.S. Fish and Wildlife Service, also Marissa Allring of the Nature Conservancy, and Jennifer Larson of the Polistes Foundation and U.S. Geological Survey. So when we started, a lot of times when people do prairie reconstruction, the practitioners think about managing invasives. This is a big thing that we do, using things like fire, herbicide, mowers, and other techniques. And when this project started a couple of decades ago, we were wondering if there were any preemptive strategies we could employ in the way we planted in order to suppress natives, increase pollinator habitat, and other things. So a primary question is, can we improve planting methods to better achieve diversity and resilience in reconstructed prairie? A basic premise of our thinking is that the characteristics of the seed mix might influence how resilient our ultimate prairie planting was and how resistant it is to invasive species. So we decided to compare plantings with differing species richness to see how things played out. Greater species richness allows a variety of root forms and above ground growth forms that could influence the success of natives, that could influence the competitive edge of the natives. And native species interactions that lead to resilience could also be happening. Phenology seems important. We wanted primary growth and bloom times to be represented throughout the growing season. We were also interested in conservatism, and that is the fidelity that a species has for a high quality natural community. So some species, when you're talking about prairies, you'll only see these species in very high quality prairies, rarely anywhere else. And those are very highly conservative species. Others are very opportunistic. They can pretty much survive anywhere. You might see them in a roadside in a brome dominated area. And those have low conservatism. We wanted to represent within our seed mix a range of conservatism. Planting methods and timing play a role in the quality and resilience of prairie. So seed rain happens naturally in the growing season, and dormant season planting allows seed to go through natural processes that facilitate germination and establishment. However, dormant planting risks seed predation by rodents and birds. The method of planting matters. Drills favor grasses, which may outcompete forbs. In some cases, planting tiny seeds with the drill can plant seeds too deeply for them to survive. Broadcast seeding delivers seed to the surface often without mechanical incorporation, which can contribute to some vulnerability, although it can also, if it's a dormant season, work in with freeze thaw into the surface layer. So the combination of planting technique and season of planting could have some pretty interesting interactions, we were thinking. So our overarching goal of this project is to use these ecological concepts to improve the practice of prairie reconstruction. And when I say prairie reconstruction, I'm talking about planting prairie from scratch, essentially, in an area where it once existed. Success can be defined in many ways. Establishment of planted species, that's a pretty basic one. If you planted that species, did that species show up? Resistance to invasives, support for other trophic levels, and ecosystem services. We looked at species cover and richness and expression of exotic and invasive species relative to natives. We're very interested in pollinators. A species rich and resilient prairie does support pollinators. It increases the nectar and pollen resources that are available and supports more specialized pollinators. The more species you have in there, the more chance you're going to be able to support those specialized pollinators. And it provides season-long bloom, especially early season, which is so important to some of our pollinators. Our objectives were to increase establishment of planted species, enhance floral resources, promote season-long bloom, and improve resistance to invasion. So in 2005, we planted and we used different methods. We did a dormant season broadcast. We tested different methods of planting, dormant season broadcast versus growing season broadcast, and then growing season drill. Also, we didn't do a dormant season broadcast or dormant season drill because it's frozen here. The soil is frozen and there's snow on the ground, and so the drill doesn't work under those conditions. We also considered various seed mixes, and if you look at the screen, you'll see that our low diversity was 10 species, medium 20, high 34, extra high was 58, and that's about as much as we could achieve 20 years ago with high quality seed and of the type that we wanted. It was just a lot of species just weren't very available. So we have planted approximately 420 seeds per square meter. As the diversity increased, the number of seeds per species declined. So in other words, if we had 10 species in our low diversity, there was the same number of seeds for each one of those diversity in that treatment, and that was repeated in the other treatments as well. Planting method and seed mix treatments were fully crossed in a completely randomized design with 12 replicates on 3 fields in Iowa. And I'm going to turn this over to my colleague Diane, and she'll tell you about what we did and what we found.
Okay, so as Pauline said, we started the study in 2005, which was the winter of 2004 to 2005, with our dormant season broadcasts. Our growing season broadcast and drill were that summer, maybe a little later than many of you would expect us to be planting, but it was a wet spring, a very wet spring. And then we monitored in the summers of 2005 through 2007, and 2010 and 2015. So we wanted to look at how things were establishing at the beginning, and we did mow during those early establishment years. And we did cover the planted species on these plots in each of those years, but only did the cover of all the species rather, but then only did the species richness on the plots at intervals like 2007, 2009, and 2015. So, to give you an idea of what our seed mixes looked like, so in our low, medium, and high, which are, I'll just call them all the lower medium, or lower richness seed mixes, we had 50% warm season grasses, which I think was pretty typical of the time. But in our extra high, we only had 13% of warm season grasses. The cool season grasses, we had 20% in those lower diversity mixes, but only 5% in our extra high richness mix. The perennial forbs, this is where we turned everything upside down. We had 68% in our extra high diversity mix compared to only 20% in the lower diversity mixes. Legumes were fairly comparable at 10% and 13% in our extra high. So you can see that we were really focusing on getting diversity of forbs rather than just diversity overall. So forbs were our focus. This is what a field would look like before we planted. This is how old corn soybean fields look basically in the Midwest. Many of you are familiar with this pattern. But they were good to plant into. You can see that they have very little, there's a lot of open dirt and places for seeds to get established. So it's actually a very good planting surface. This isn't at Neil Smith, but just to give you an idea of what the fields looked like when we established our experiment. Each of these blue boxes is a treatment cell. The treatment cell is where we applied the species mix and the planting method. So since they were fully crossed and there were 12 reps per treatment, we had 144 different treatment cells at each field at Neil Smith Refuge. And so the tractors came in and applied these plantings in a completely randomized design across the cells that you see here. The treatment cell is large. It was about 12 by 12 meters. Where we actually sampled was in the middle of that treatment cell, and it was 2 by 6 meters. So there's a difference. We just tried to stay in the middle because, edge effects, you might get some little spillover or what have you. So the center was where we were sampling. So getting right into each objective, I'll give you a summary of the results for each of the objectives that Pauline talked about. First of all, when we planted more species, we got more species. That's very reassuring, right? So as we had, you'll see here, the x-axis here is the seed mix richness. This is the mean number of species per plot. And so at low, we had more or less 1 species per plot on average. And our extra high, we had over 8 species per plot. And these were all statistically significantly different from each other. Cover, again, going from low to extra high. And this is cover as a proportion of total live. We did that because we had comparisons that went across seasons, or years. So we wanted to not be showing you year effects when they were really not part of the things. We just didn't want to tell you about year effects at this point. So basically from the low seed mix richness to the extra high, we had increasing cover, but the high and the extra high had similar cover. And these are forbs, okay? So again, we're talking just about, did we get the forb richness that we were hoping for? And we did, and we got good cover of those forbs as well. So then Pauline mentioned the coefficient of conservatism. And I think you probably all have heard of that, but just to emphasize, that's just an index from 0 to 10, with 0 being a weed that's not even native, and 10 being something that's absolutely restricted to high quality prairie. So looking at the average coefficient of conservatism, starting in 2007, when we first did our first survey of complete richness, the low diversity mix had far lower coefficient of conservatism than any other mix. When we got into 2010 and then finally to 2015, we had our extra high diversity mix pulled away. And you see that there was higher coefficient of conservatism as we increase the diversity. But what you also see is that we're getting an increase over time, even in our low diversity mix. And what that's showing us is that we're having seed dispersal from our higher quality mixes, our higher richness mixes into these lower richness mixes, which is, I think, a pretty important thing when you're talking about how much money do I have to spend on seeds. If they're going to disperse themselves, that's kind of a, that's a boon. Another objective was increasing establishment of planted species. So what I'm showing you here on this slide is that in our low diversity mix, 10 species, we got 100% of those 10 species. But remember, we had a lot of seeds per species in that mix as well. So part of it is just, how many seeds, what was the propagular pressure? And it was higher in the low diversity. 95% of 20 species established in medium, 85% of 34 in high, and 75% of 58 species in the extra high. Now, it's clear that in the extra high, we still had more species, but as a proportion of what we planted, we had somewhat fewer, but we planted some really highly conservative species, and not all of them have come up yet. And maybe because we were seeing that increase over time, perhaps we'll still get a better establishment in the long run. But at this point, you have to kind of prepare yourself, and especially if you have volunteers working on your sites, just prepare them that not everything is going to be visible at first, even within 10 years, and this is at the 10-year mark. So we also wanted season-long bloom, and I wasn't out there counting flowers. So what this is showing you is the potential bloom based on the left here, the seed mix, and then on the right, what species we had after 10 years. Okay. So for what we planted, we only expected to get April bloom in our high and extra high diversity mix. And we expected pretty high amounts of species that would be in flower in the middle of the season in the extra high mix. And then what you see here, what we got, because of dispersal out of these higher diversity mixes into the lower diversity mix, we had April bloom in every diversity mix that we had out there. But we had fewer species that actually established in the middle of the season. So that's unfortunate, but at the same time, that's not bad. And the fact that they were spreading over the entire field as opposed to just in their little spaces, I think that says volumes about the potential of species to move around. And then we also had really pretty good bloom in our October at the end of the season. So then finally, this is the last thing I'll be talking about, resistance to invasion. This is, if you look on, this is year on the x-axis and exotic cover as a proportion of total live on the y-axis, and our low, medium, high, and extra high. And okay, in 2006, 2007, early on, we had differences, and we actually published on this and said, gee, there are differences in exotic cover, but by 10 years, there were no differences anymore. And I think that's important, just because patience can pay off. You wait, you don't get excited, you don't go in there, we didn't spray, we didn't do any of that kind of thing. This is just what happened based on the species interactions. And we had that cover show up as insignificant. The other thing, however, the devil's in the details. So here we have, these are a lot of small pictures, and I apologize for that, but it was important to show them all to you at once because we have Canada thistle, Queen Anne's lace, musk thistle, and curly dock. Okay, those are all the Forb species that we have as invasives and that are troublesome in our area. And you can see that the extra high diversity mix is this filled dot. And by the end, these were all, the filled dots were among the lowest cover values that we had. Now look at smooth brome and Kentucky bluegrass. These are invasive cool season grasses. And what you see is that unfortunately, we have an increasing trend across all of our treatments, but the extra high diversity mix has the highest mean cover, the greatest increase in cover over time. And that's what really is going to be the most threat to these fields, these prairies, is just being taken over by these cool season grasses. And now we go back to Pauline.
Okay, sorry, my slides weren't advancing there. So I want to talk about what we learned. And one thing that we learned is that forbs disperse into lower richness plats. And this is something Diane pointed out in the data slides. I mean, this was especially evident in the 10-year monitoring event. And as she mentioned, it's important to understand that some of these things happen through time. We learned that proportionally species richness declined with increased planting richness, but mean species per plat increased. So in other words, in the low diversity, we only planted 10 species and we observed all 10. In the extra high mix, which was 58 species, we observed 75% of the total species mix. However, we observed more species richness in the extra high plat. So although we would have liked to have had 100% of the species expressed in that extra high mix, we still achieved a measure of success that would have otherwise been impossible. So a lesson is, if you want the species to get out there, you need to put them on the ground. And that sounds really trite almost, or simple, but you know, even if there's a few that establish of that species, you have picked up more species diversity in that plat. We learned that mean C was still increasing at 10 years. So especially in the low and extra high mix, we're still getting more species increasing in those species, in those mixes on the plats. The low species mix gains species from the higher species richness mixes, and new species are still appearing in the extra high mix. And one of the things that Diane said is that it takes time before some of these things get observed. And I might also say that it's possible that some of the species that that took a little bit longer to show up in the monitored subplots actually came up in the larger plots. We remember we were monitoring a smaller area in the center of a larger planting plot. And so it's possible some of those were there, but we were monitoring only in the center. Invasive species, especially cool season grasses, are the biggest impediments to resilience in this experiment. One of the things we have to remember is that the conditions that surrounded this experiment certainly do influence the results. Long-lived cool season graminoid competitors were sparse in all of our mixes. That was not because we ignored that need to put those cool season species in the mix. It was because they really weren't available to us, particularly 20 years ago when this started, the types of seed and the quality that we wanted. So we did put in Canada wild rye, which did very well, and it did suppress weedy species, but it didn't, once it was burned, it has a genetic predisposition for decline after fire. So that's what happened. And our longer lived, we just didn't have enough longer lived species in the mix. The ones that we had, particularly Stipa in Iowa, Stipa spartea, needlegrass, didn't really do very well. And then so one of the things that that we would encourage you to think about is to consider including a component of sedge and cool season graminoid species. I would also say don't rule out forbs, but I think the graminoids are important for competition with those cool season exotics. And it's also possible that if we had burned more, that those cool season exotic grasses may have had a little harder time. We also learned, you know, you can be strategic in the way you plant your prairies. Because, as Diane mentioned, we were finding species showing up from different mixes. We know that seed was moving around, and that we were gaining species in some of the mixes from other higher diversity mixes. So, you know, you don't have to plant everything in one spot. You could think about planting a mosaic on your planting field. You could plant things in whatever pattern makes sense to you. Some people might do it sort of along topographic lines, or maybe in straight lines to accommodate a particular kind of planter. But you can make that, put multiple mixes on a planting site. And through time, you could expect that you're probably going to get more species, you know, that species mixing throughout the field. If you have expensive or hard to get species, and you don't want to just throw them out in the general mix, it's another approach could be that you plant a basic prairie mix, and then you put in islands of these expensive or hard to get species within that, dotted throughout that basic mix. And we could expect that there's a high probability that you would start getting, through time, some of those species to be coming up and infecting and spreading into the lower diversity mix, or the mix that is a more generalized mix. And then finally, you know, these things weren't all showing up in the first year. We're still getting more species expressed. So we know that those species are moving around from seed rain and from the species that were already on the plots. So you could do that seed rain. You could make more species available in a planting by simply going out and seeding diversity, greater diversity through time. So you could phase in your the species that you don't have access to as easily through time. That would be another strategy. Now this slide isn't a part of what we have done, but it's about a next step that Diane and I are pretty excited about. I wanted to make the point that we don't know everything about the conditions that influence a prairie planting and how it succeeds in any given experiment or time phase. But you know, one of the things that we know that isn't getting looked at enough is mycorrhizal fungi and soil microbial communities, and those affect the way things turn out. So that could be an important next step. So I'd like to acknowledge the U.S. Fish and Wildlife Service, Neil Smith National Wildlife Refuge staff and volunteers, and U.S. Geological Survey Science Support Program, and the Northern Prairie Wildlife Research Center. Many people have contributed to this. So thank you. And if you have questions, we can entertain questions.
Yeah, there are lots of questions. So one of the first questions came up around soil prep, and then matching. Did you do any matching of seed to soil type?
No, we did not. It was a matter of trying to we didn't match seed mixes to soil types.
Another question was around the extra high diversity mix. Was it allowed to seed in low diversity plots?
Yes.
Okay.
Yeah.
We didn't do anything to stop natural dispersal at all.
And another question.
Yeah, these fields were just out there in that. Go ahead.
Go ahead. No, answer.
No, these things were, if you remember the slide that Diane showed you that had all of those lines with the plots kind of denoted, that's the arrangement that they were, and there wasn't any kind of management that would favor one thing over another.
And one question that came up was, have we analyzed that sort of spatial orientation and how that affected, and we have not. That I think it would be kind of messy that between those rows of cells, there was just a sort of a grass mix that a native grass mix that we put in. It could happen, but I think there's enough confounding that it might be very difficult and not very convincing.
Also, there's one question. Do you have any thoughts about the value of waiting to seed more conservative species for several years after the initial seeding?
We haven't done anything with that. There are various conditions, I think, that will favor interseeding as opposed to not favoring it. If you're interested in talking about that, I can talk about that a little bit, but you need to have places where there's soil showing. Certainly, if the seed doesn't hit the ground, it doesn't grow. But we haven't worked on that.
We have another question. Please address what control you might have done for thistle, Queen Anne's lace, parsnip. And did you say you had no control?
We did no control at all, aside from the mowing that we did in the first couple of years for all those annuals that come up. We did no control.
And
I should point out.
Go ahead.
I should point out that we did do 2 burns. It wasn't necessarily for control of those things, but it was a part of the system. So within the 10 years, there were 2 burns.
And some just wanted to be reminded, what was the ratio of the grasses to forbs in the extra high mix?
We had 20%. Let me look, just make sure I don't say the wrong thing. Go ahead and I'll come back with that information.
Then another question is there a trend of increased cool season grasses likely related to reduced presence of warm season grasses in the higher diversity mixed units?
I guess I would, my response to that is that no, it isn't reduced warm season grasses. It's lack of cool season natives.
And did the inverse relationship between seed mix diversity and establishment of species indicate competition within the seed mix?
People have suggested that could be the case. We don't have any, we can't test that with what we did, but it's certainly a possibility.
Okay.
But we did have the same number of seeds total, but if we had more of one that was especially competitive, that could have made a difference. And just to go back to the previous question, we had 13% warm season grasses versus 68% forbs in the extra high.
And just as a comment, the kind of seed mixes that we use for this experiment were to test specific situation. And if I was planting seed mix on a piece of ground, these are not the kind of seed mixes I would use. I wouldn't try to make the same number of seeds for every species. I mean, if you look at something like compass plant, for example, or indigo, let's say the false or white indigo, those species, if you plant 10 of those in a relatively small area, or you plant 10 yellow stargrass, which is a very diminutive plant, you're going to have very different sorts of results. And if you plant 10 Monarda fistulosa, the horse mint, you're going to have a very different sort of relationship to the species with one another, and they just grow different. They occupy different sorts of niches. But for the purposes of this experiment, we really wanted to test things in a way where we had sort of a constant number of seeds for each species.
Did you use local source seeds or cultivators?
These were local. These were for Iowa, for all of the things we're talking about here. This was local ecotype seed, and there was according to the Neil Smith National Wildlife Refuge local ecosite type seed requirements.
We have a question. Do you have any recommendations for site prep for former pasture land plantings?
Well, for pasture land, you know, site prep for something like that, various people will tell you different things. I think the main thing is if I have some land I'm looking at right outside my window where I live, and it was a chunk of it was just brome. And, you know, what I have found is that burning it annually reduces the success of the brome, and I interseed, I just throw seeds out onto the ground in the winter or in the spring early, or even in the fall. And they tend to come up fine in that brome that has been, and other cool season exotic species that has been decreased in vigor by the burning process. There's a number of things you can do. The main thing that you want to have is, like I say, soil showing, and allow those species to be competitive.
Any guidance on the importance of local provenance seeds?
Yeah, and I saw a yes come up in the chat somewhere, so I assume that was related to that question. And, you know, I guess it depends on the species, I think, you know, and this certainly wasn't a part of what we were testing, so we can't answer that from a scientific perspective from this project. But I think it's very important for some species, perhaps a little less for some others. You know, you may have a wider range for some than for others. And I know in Minnesota, there was, I don't know, 30 years ago, maybe, some seed planted that was intended to be used for dense nesting cover for waterfowl. And they thought they would harvest the seed and move it to another place. Well, they got the seed from Nebraska, and it did great. It grew. It never produced a single seed that they were able to harvest. And so, you know, all that does is illustrate that it isn't the same place where it evolved. So there's local variation in terms of how things work. I think it's important. We could get into this a long discussion, so I'm not going to take any more time on that.
What is the best way to deal with foxtail? Any suggestions?
Plant perennials. You know, that's many of the foxtails are annuals, not all are, but they don't tend to compete well with a good, well-developed perennial vegetation.
Did you use any references for the prairie community or literature as to natural community makeup in regard to designing your mix?
We did, in terms of, we were thinking of the guilds that Diane mentioned, and we were interested certainly in, you know, like I say, that balance of conservatism, and that comes from the literature about conservatism, you know, and making sure that if you have things that are very highly conservative, those are included, as well as those that are poorly conservative. And so that gives you a range of sort of responses by the plant species to one another and to the conditions that they find. I guess that's one answer. We did try, yeah, I'll leave it there. Diane, if you have other thoughts.
Also, Diane, you did mention a publication. If you could put the name of the publication or a link in chat, I think people would appreciate being able to refer to it. Was the data for dormant broadcasting analyzed separately than growing season seeding treatments? So did you analyze the dormant broadcasting?
They were analyzed in a, I'm going to say this wrong, generalized linear model. So they were treatments within a single model. Does that answer the question?
Some more questions on invasives. Do you not have, and I'm going to mutilate the Latin here, Lespedeza cuneata or bird's foot trefoil? Because I think you said you didn't spray for invasives.
We did not, and yet at Neil Smith Refuge, there is Lespedeza cuneata. It did not show up in our plots. And so when we talk about invasive species that are very problematic, I mean, that would be one of the ones that I would put kind of up there to pretty top of the list, that suite of species that's really very difficult to control. But we didn't have it in our plots. Yes, there is Lotus corniculatus, the birdfoot trefoil is there, and there's a number of the usual cast of characters that you would be concerned about with prairie reconstruction, yeah, on the refuge.
And we've had a couple questions. You may have mentioned this briefly, but people wanted to know more about your planting methods, broadcast, drill, et cetera. Did you find one to be more successful than another?
I'll take a first stab at that. Initially, it seemed like we had more forbs coming up in the broadcast, which you would expect. And that generally we had greater establishment of the warm season grasses in the drill, which you would expect. By 10 years after, most of those those differences really were not that strong anymore. They were all about the same. I guess the big difference that probably persisted was that we saw that increase in the invasion of the cool season grasses in our low diversity treatment, but that wasn't affected by how we planted the dormant versus the drill. Initially, our recommendation after 5 years was don't do a growing season broadcast. It seemed to be the worst. It seemed to have the most weed pressure. But at this point, it didn't make any difference after 10 years. So there's just a lot happens over 10 years that sort of equalizes things. And I don't know what all that is, but if I had the opportunity to do a planting, I'd probably just take that opportunity and I wouldn't worry too much if it was dormant or growing or what have you.
Mm-hmm. And I think also I would add to that some of the things that we couldn't really test were the effects of weather conditions at a particular time in the process of that planting. And I think those kinds of things, if we had in the early 90s a long stretch of really intensely wet years, and there was a period of time where there were some very dry years, and I think the way those things interact with each other influences the way prairie plantings turn out too. And so I don't know, what if we'd had different weather conditions at different points in time on this? I don't know if it would change it or not, but I think Diane's point, the data bear it out, is well taken.
Can you remember some of the highly conservative species you planted that did fail to appear, or conversely, were there other conservative species that did particularly well?
Yeah, I remember looking this over, and Diane, I think lead plant was one of them. Is that correct?
Lead plant really didn't come up at all. We had no lead plant. I'm looking back in the paper.
Yeah, I'm trying to pull it up.
Came up pretty much everywhere, and it's an 8. Helianthus occidentalis was sadly absent except in the extra high for the most part, and that was a bit surprising. Dahlia candida did come up everywhere, although not strongly.
Sprablus heterolepis, the prairie drop seed, that was another one.
Yeah. Oh yeah, the heuchera did not. Heuchera richardsonii only came up in a small amount in the extra high diversity, which was where it was planted. but it didn't disperse anywhere else and came up sparsely where it was planted. In the paper that this talk was based on, we do have the list of all the species where they were planted, their coefficient of conservatism, and how they were doing by the 10th year. So if you want to take a look at that, you can look at more details. And it's actually in the paper, it's not supplemental. Yay, NAA.
And yeah, and I love this table that's in this paper because it's not just about the conservatism. It's really a summary of pretty much everything, everything we did, and so if you're interested in those kinds of details, like you were just asking about, I highly recommend you get to this paper and take a look at that table.
Okay, we are just at about 5 of 1, so I'll ask 1 or 2 more questions and then we'll wrap up. Was population of pollinators monitored as part of the evaluation?
If only. No, that would be a great thing. But you saw the way the plots were laid out. I think our treatment cells were too small to really separate treatments by who was visiting because of the spillover effects. But other people are working on that right now. Ian Lane has some great data on that. So, it's being done, just not by us.
Mm-hmm.
So we do have one comment, Dr. Drobney. She loved your America's Lost Landscape.
Oh, wonderful. Thank you.
Did you consider using container plants, however limited, to introduce those species that didn't do well from seed?
No, this was not what we were testing. We weren't trying to find out. That would have added another whole, confounding thing to our study. So we didn't do that in this study.
Okay. All right. Well, I think we have to wrap up, though there are more questions. This has clearly been a popular topic, but I want to thank you both today for being here. We really enjoyed your presentation and hope to see more. If you don't mind sharing a link to the paper, if you can't do it in chat, send it to me and we will post it in the webinar archive so that it is available to people who want to see that. And then to all of you who joined us today, I want to thank you and invite you to come back in February and see Project Budburst. And we look forward to seeing you then. So thank you, everybody. Have a great day.
Thank you.
Bye.
Thank you.